in the University of Illinois as a graduate student, he explained to me that one of the things that attracted him to fluorescence was um, the fact that it was not only allows you to learn a lot of information at the molecular level, but it was aesthetically pleasing. And we should always enjoy the colors involved in fluorescence when we have the opportunity. Certainly, fluorescence can provide information on molecular environments. In fact, fluorescence probes have now been developed, as you'll learn, that can probe almost any chemical or physical environment, such as viscosities, polarities, pH temperatures, et cetera, et cetera. And you'll learn about many of these probes. And what's of particular interest to me during my career is fluorescence provides information on dynamic processes on the nanosecond timescale. You can think of molecular probes essentially as molecular stopwatches. And the stopwatch starts when you absorb a photon of light and stops when you emit. And by attaching the stopwatch to molecules of interest, you can learn information about dynamic events that occur during the lifetime. So, coupled with modern fluorescent microscopy, confocal multiphoton that you'll learn about, and fluorescent proteins, such as green fluorescent proteins and others, it's providing now extremely detailed spatial information in living cells, but also information on the dynamics of the cellular components, which I think is one of the great aspects of fluorescence microscopy. And it's also very, 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 many more varies, sensitive. So work with nanomolar concentrations is routine, and femtomolar, even single molecule studies are possible with some effort. Well, what are the experimental systems you can look at? Well, any biological system virtually, from protein structure and dynamics, membranes, cell organization and function, and you'll learn a lot about that in this course. Engineered surfaces for high throughput drug discovery, and live animals. What type of instrumentation is used? Well, we have fluorimeters and fluorometers, and we'll learn the difference. Of course, many types of microscopes that you'll learn a lot about in this course. High throughput plate readers and intravital imaging, and you'll learn how you can use these modern techniques to go deep into biological tissues. Now, virtually all fluorescence data required for any research project falls into one of the following categories: the fluorescence emission spectrum, the excitation spectrum, the quantum yield, the polarization or anisotropy of the emission, and the fluorescence lifetime. And in this lecture, we're going to examine all these categories, except lifetime, which will have its own lecture, and discuss some of the underlying concepts and practical considerations. And a good place to start is always the Perrin-Jablonski diagram. Some people call this the Jablonski diagram. Many of us prefer to call it the Perrin-Jablonski because Francis Perrin, you see, contributed to this before Jablonski even. But at any rate, this diagram is useful to consider sort of the life history of these excitation processes. So molecules can sit without perturbation in the ground state, and that's the S0 state shown here. And there's different vibrational levels in each of these different electronic states, and normally at room temperature, molecules would be in the ground or the lowest vibrational level. And upon absorption of light of whatever frequency it absorbs at, you can go to different excited states, different vibrational levels in those excited states, is shown here, S1 and S2. But regardless of which excited state you reach, you have very rapid internal conversion in the order of picoseconds, where all the molecules, the energy is redistributed, and you end up at the lowest vibrational level of the first electronic excited state. And there you sit until something happens to deactivate it. In our case, we hope it's fluorescence, which takes place in nanoseconds. You can also have radiationless decay that brings it down to the ground state. You, in some cases, can have intersystem crossing to go over to a triplet state, which can lead to phosphorescence. We're not going to worry about that here. And what's very important for many of the lectures in this course, you can also deactivate the excited state by passing the energy to another molecule, an acceptor, in the process of Förster resonance energy transfer, which you'll learn a lot about. So in a typical emission spectrum, the excitation wavelength is fixed and the fluorescence intensity versus wavelength is obtained. And it used to be we could only do this with cuvette studies in vitro. But now, as you'll learn, there's microscopes that allow you to get the emission spectrum also, which is very important for some of the studies you'll learn about. Here we see four different fluorophores: quinine, fluorescein, rhodamine, and bromide, and their emission spectra. And I'd like to show you what these solutions actually look like under UV hand lab. And when you've been in the field as many years as I have, and Enrico also, of course, you can look at these colors under the hand lamp and judge within a few nanometers usually where the emission maximum is. You'll just have to trust me on that. Now, examination of a large number of emission spectra resulted in formulations of certain general rules over many years. So for a pure substance existing in solution in a unique form, the fluorescence spectrum is invariant. That is, it remains the same independent of the excitation wavelength. It's very important for many of the things we do. It also lies at longer wavelengths than the absorption. And to a good approximation, the fluorescence spectrum is a mirror image of the absorption band of least frequency or highest wavelength. And we show that in this diagram here. So this is looking at fluorescein in an alkaline solution in a cuvette, scanning the emission spectrum. And I show on the left the excitation at 280 nanometers, 375, and 470. We can see when we excite at 470, we get the largest spectrum. That's because it absorbs more at 470 than the other wavelengths. But also these raw, uncorrected spectra will depend on how much exciting light comes out of your excitation source, in this case, a xenon arc lamp, the monochromator efficiency, etc. But we notice when we go to the right, if you normalize these all on the computer to the same maximum intensity, they're overlap. And this demonstrates, yes, regardless of where I excite, the fluorophore, the emission wavelength is always the same. Okay. And what about the shift? Well, here's a typical absorption and emission spectrum of fluorescein. You see the absorption in black, and we see the emission in green is shifted to higher wavelengths, lower energy. And this is called actually the Stokes shift, and we'll learn in the history lecture a bit about George Stokes, who first observed this phenomenon. And finally, maybe less important but of interest, is the here we see rhodamine absorption spectrum, the solid line, and the dotted line is the emission. And yes, to a first approximation, it's the mirror image, the emission and the absorption. And these rules follow from the Perrin-Jablonski diagram we discussed earlier. Here's an abbreviated version of it. Specifically, even though you excite to different electronic levels and different vibrational levels in these electronic levels, the emission always comes from the same lowest vibrational level of the first electronic state. So that's why the emission is independent of the excitation wavelength. And the ground state at room temperature, the vibrational levels, you're normally in the lowest vibrational level. So you're always starting from the same point, but you can go to higher levels, taking more energy, so at lower absorption wavelengths. But the fluorescence is always from the same part, and the fluorescence can go down to different vibrational levels. So if it goes to a higher vibrational level, of course, it will have a lower energy. So that accounts for the Stokes shift. And the fact that the spacings of the vibrational manifolds in the ground and first electronic state are usually similar. It accounts for the approximate mirror image relationship. Now, what about the excitation spectrum? Well, to do that, that's the relative efficiencies of different wavelengths of incident light to excite the fluorophore. So in a spectrophotometer, you would scan the different exciting wavelengths and keep the emission wavelength constant. And if the system is well behaved, that is, if these three general rules we outlined hold, one would expect the excitation spectrum to match the absorption spectrum. And here you see the absorption spectrum and corrected excitation spectrum for analytical naphthalene, sulfonate, and ethanol. And you see they overlap very well. I say corrected because there's a lot of instrument correction factors before you can get these excitation spectrum. We're not going to go into that in this lecture. Also, though, I want to point out, you may say, well, why do I need excitation spectrum if I've got the absorption spectrum? Well, a couple of reasons. First of all, you can get this excitation spectrum at much, much lower concentrations than you could ever get the absorption spectrum, because fluorescence is so many orders of magnitude more sensitive than absorption. But also, it's of interest, if the excitation and absorption spectrum don't match, then things get really interesting. For example, consider the excitation spectrum below for a fluorophore-free in solution. Then we're going to see bound to a protein, what could happen. The emission depends on the fluorophore, of course. In this case, I'm looking at danzo, naphthalene group, and the emission would be about 470 nanometers, let's say. And this is free in solution, the excitation spectrum, which would match the absorption spectrum. But if we do the same experiment when this danzo is bound to a protein, you may see something like this. And we see that there's a big deviation down at lower wavelength, 280, for example. Well, that's because you have energy, you can have energy transfer from the protein's tryptophan residues to the bound fluorophore, resulting in an altered excitation spectrum. And also in the case of FRET, you would get alteration. So these changes in excitation spectrum can be very important for pointing out some underlying photophysics in the system. Now, the quantum yield was a concept introduced in 1924 by Sergei Vavilov, and the simplest definition is that it represents the number of photons emitted divided by the number of photons absorbed, as we see here. And notice you can't say the energy emitted over energy absorbed because of the Stokes shift. They always have different energies. So, for example, if a fluorophore absorbs 100 photons and emits 100, the quantum yield is 1 or 100%. If it absorbs 100 and emits 20, the quantum yield is 0.2 or 20%. But a more insightful definition of quantum yield would be that the quantum yield equals the rate of the emission process divided by the sums of the rates of all other deactivation processes. So here we have Kf, the rate of fluorescence, and the sum of Kd designates other rate constants that depopulate the excited state. These can be various things: photochemical, quenching, dissipation of the energy as heat, or also FRET. Now, I think it's interesting to look at quantum yields for different compounds, and here's a list from Bernard Valleur's excellent book, Molecular Fluorescence. And we can see from some like benzene, it's pretty low, and we go tryptophan, anthracene, quinine sulfate, a popular standard, up to rhodamine, which has a quantum yield of 1, so it's perfectly efficient. But I want to point out that really, whether we consider a fluorophore depends in large part on the quantum yield. What I mean by that is some molecules like adenine, guanine, adenosine, these things that make up DNA or an RNA, for example, they do fluoresce, but they fluoresce, the quantum yield is so low, 1 photon emitted over 10 or 20 or 30,000 absorbed, that they have no practical value as a fluorophore. So really, when we talk about fluorescent molecules, we're implicitly saying something that has a decent quantum yield. And I also want to point out that quantum yields are notoriously difficult to measure. And I don't care if an instrument salesman comes to your lab and say, our machine can do this really easily. That's not true. Reliable literature values for quinine sulfate, for example, from good labs have been published ranging from 0.5 to 0.7. So what I care about if I'm reading a paper where the authors give the quantum yield is how did you do the measurement? What was your standard and what value did you use for the standard? Because if I think your standard should have a different value, then I can substitute. So what about polarization? I'm going to talk a bit about that, although you're not going to really discuss polarization in this course. But I think it's important that all practitioners of fluorescence be aware of the basic principles, because it's a very, very, very popular technique in the literature, polarization or anisotropy measurements. And it behooves you, it's good for you to know the basic principles. So let's start in. So we know that light can be considered oscillations of electromagnetic field characterized by electric and magnetic components, which propagate perpendicular to the direction of light propagation. Now, in these lectures, we're only going to consider the electric component. Now, in natural light, the electric vector can assume any direction of oscillation perpendicular or normal to the light propagation direction. I try to illustrate here. And polarizers are optically active devices that can isolate one direction of the electric vector. So here we see a natural light source with unpolarized light. We put a polarizer and we get one plane of polarization. One electric vector is selected. If we rotate the polarizer, we rotate the direction of polarized light that we're looking at. Now, polarizers come in different types. In a good spectrophlorometer or fluorimeter in the lab, you usually use calcite crystal polarizers, which are very expensive, but they can go down in the deep UV, and they're among the best. Very popular to also have less expensive polarizers, these sheet polarizers, which are very good, but they're not as good when you go in the lower wavelengths. Now, of course, being in Hawaii, it's very important for me to have my own polarizers, my Maui Jim sunglasses. And many polarizers, I'm sure some of you have them, are polarizing sunglasses. Many sunglasses are polarizers. And I want to ask you, and I'm not going to tell you, but you can think about it and can't get the answer, ask me later in discussion. What direction of polarization do sunglasses pass, the vertical direction or the perpendicular direction? So think about that. Now, consider an XYZ coordinate system where the fluorescent solution is placed at the origin. So as we see here, X and Z is in the plane of the page, and the observation is made in the Y-axis, so out towards you and the audience looking in. So you can have exciting light traveling along the X direction. You could put a polarizer and isolate one electric vector. In this case, I'm showing one parallel to the Z-axis, which corresponds to the laboratory vertical axis. Now, this exciting light will be absorbed by the fluorophores and give rise to fluorescence. And then to look at the polarization of this emission, we put a polarizer along the emission pathway and we rotate it, and we can view how much of the intensity is parallel and how much is perpendicular to the vertical laboratory axis. Polarization is simply defined as the parallel intensity minus the perpendicular over the sum of these intensities. But it's important to know about another term, anisotropy, that you see very, very often in the literature, usually designated as A or more commonly as little r, which is defined as the parallel minus perpendicular intensities over parallel plus twice the perpendicular. And we won't go into the reasons for this, but that's the definition of anisotropy. Now, let me make a comment about the difference. So you can show, given these definitions, that anisotropy is 2/3 times the reciprocal of 1 over polarization minus 1/3. Or more simply, anisotropy is 2 times P 3 minus P. For example, if you have a polarization 0.5, the anisotropy is 0.4, 0.1, 0.069, etc. And usually in modern instruments, when you're measuring polarization, it will automatically give you their anisotropy, so you don't have to calculate it. Now, clearly the information content of the polarization function and anisotropy functions are the same, and the use of one term or the other is dictated by practical considerations. And we might mention some of these as we go along. Now, when we excite an ensemble, a collection of randomly oriented fluorophores with polarized light, we're performing a photoselection process, selecting a population of excited molecules that nominally, that is more or less, have their excited dipoles lined up with the polarization direction of the excitation. This process is illustrated here. But let's look a little more closely. What actually happens when you absorb a photon of exciting light is you create a dipole moment in the fluorophore. Now, fluorophores like the one shown here, which is Laurdan, you'll hear about this later in the course, you can have groups that can stabilize, in this case a carbonyl group can stabilize partial negative charge, or an amine that can stabilize a partial positive charge. And in the excited state, then, the electron cloud is dispersed to form this dipole in the molecule. And this is the transition dipole or transition moment. So when we're exciting with vertically polarized light, in this case, we excite molecules that are more or less lined up with that direction. What do I mean by more or less? Well, in fact, the probability of absorption, of course, it depends on the extinction coefficient of the molecule, et cetera, but also depends on it's proportional to the cosine squared of the angle between the exciting light and the transition dipole. Okay, so that if the exciting light is perfectly aligned with the absorption or transition dipole, the probability of absorbing is 1, cosine squared. Even at 45 degrees, you see there's a decent probability, 0.5, of absorbing. But at 90 degrees, when the absorption dipole is perpendicular to the exciting light, it will not absorb. And let's illustrate this with this little cartoon. Here we have a bunch of molecules in the ground state, and that's the direction at that instant of time what their absorption dipole, transition dipole would look like on the molecule. Now we excite with light, vertically polarized light, and we see the bright circles now correspond to the molecules whose dipoles are more or less aligned. And you see they can be a little off. The dimmer green circles are those that still absorb, but they're at a larger angle, so they don't have as high a probability. And the black circles are those molecules that the dipole was at perpendicular, so they don't absorb at all. So this is really the photo selection I'm doing. Okay, and first we're considering consider the case where the excited dipole is parallel to the emission dipole, okay? And let's also consider the molecule cannot rotate during the excited state. So we used to do this by putting the fluorophores in a highly viscous solvent at very low temperatures, like glycerol at minus 50 degrees centigrade. And I won't go through the math because we don't have time, but in these cases, then if the excitation emission dipoles are lined up, the highest polarization you can observe will be plus 1/2. And if they're perpendicular at 90 degrees, the polarization you would get would be minus 1/3. So the limits of polarization are plus 1/2 to minus 1/3. And that's important to know those limits. I see over the years, many students I noticed would just assume, or researchers, not just students, that the polarization, the maximum is 0.5. Well, not quite. In fact, the limiting polarization, which we call P0 in the absence of rotation, is related to the angle between absorption and emission dipoles by this expression. So This allows you to understand that limiting polarization of fluorophore will depend on the exciting wavelength because it depends which absorption or transition dipoles are excited, if those dipoles are lined up or if they're at a large angle. For example, this is what we call an excitation polarization spectrum for tryptophan or indole, virtually the same. And we see the dotted line as the absorption spectrum, and the solid line with the points are the actual measured um, polarizations. This is for indole and glycerol at very low temperatures. And we see it's very complex because of multiple overlapping absorption patterns. This measurement was actually done by Bernard Verloor in Gregory Weber's laboratory many years ago. Now, let's look at another molecule, rhodamine, B, embedded in a lucite matrix. So this is a room temperature, but it's in a plastic matrix. It can't move. And again, we see this variation of polarization depending on the wavelength. We see at wavelengths above 470 nanometers, you get up to nearly to 0.5, the theoretical limits, but then you can get some very low polarizations below minus 0.2, for example, when that absorption dipole is at a large angle relative to the emission. Here's GFP. I'm sure many of you probably never seen this. I do this in my lab where you normally excite, of course, in the visible where you have a high polarization, not 0.5, only about 0.42 or something, but you can go lower. So why is it important to know these excitation polarization spectra? Well, so you can choose the best excitation wavelength, of course. So if we go back to tryptophan, maybe you're studying a protein. And many people studying proteins like to excite the protein with 295 nanometers because tyrosine no longer absorbs at that wavelength. And they say, well, then I can look at the tryptophan in isolation. That's fine unless you're doing polarization. As you can see, then you're going to be near the minimum value. So if you're doing polarization studies on a protein, it's better to excite near 300 nanometers. And this has become particularly important in the last couple of decades when many people use laser sources. And obviously the lasers will have different and fixed excitations. For example, if you use the helium cadmium laser, one wavelength that it emits at is 325. If you happen to do that in GFP, you would get a low polarization. Of course, with an argon ion laser at 488, no problem. But that's why it's nice to keep that in your mind, what these values are. In the case of multiphoton excitation, the limits are different because you're getting essentially a selection process on top of a selection on top of a selection, as shown here. So in the case of one photons, the limiting polarization is 0.5 or anisotropy 0.4, which is 2/5. We see the R0 is 2/5. For two photons, R0 is 4/7. For three photons, you can even get up to 2/3, 0.67. So you have some advantage for doing polarization with three photons, or multiphotons, I should say. Now let's consider what happens when the fluorophore can rotate during the excited state lifetime, because the fact is many of you probably aren't interested in only looking at samples in glycerol at minus 60 degrees centigrade. Well, we might have the absorption dipole, and the emission dipole may already be at a slight angle. But during the excited state lifetime, we can have rotation of the molecules, so the emission dipole can change. And in fact, you get additional depolarization occurs if the dipole can rotate through an angle omega. And then the observed polarization, P, is related to the limiting polarization. Of course, that's always your starting point at the wavelength you're using, times 2/3 cosine squared omega minus 1. So you observe the polarization, and knowing the P0, you can see what angle it went through. For example, if we have a distribution in solution, we excite with polarized light, we do our photo selection. Now we can have Brownian rotations of the molecule during the lifetime of the excited state. And Francis Perrin related this already in 1926. And we rewrite his original equation in terms of polarization measured, P0. Lifetime, of course, the lifetime is important. The longer the lifetime, the more time it has to rotate before emission, and the birotational relaxation time, rho, which is the time for a given angle to rotate, a given orientation of a molecule to rotate through an angle given by the arc cosine of 1 over e, 68.42 degrees. Now, if we have a spherical molecule, rho 0 would be equal to 3 eta, which is the viscosity, V molar volume over gas constant temperature. For a spherical protein, it follows then you would have 3 eta, m the molecular weight, times the partial specific volume and the degree of hydration. And that's fine for what you would expect for a protein. And of course, if you have ellipsoids or other shapes, it will be different. And that was studied in detail by Gregorio Weber, as we'll mention later. But it's also important for us to know that eta is the viscosity. So yes, the more viscous the surroundings are, the slower the rotation. And that's relevant for membrane studies, as we'll see later. So if a fluorophore is free in solution, it could have a fast rotation, which would give a low polarization. It's bound to a protein, for example, a macromolecule could have a slow rotation because of the slow tumbling of the protein and a high polarization. I want to also point out that in the literature, you'll often see a term rotational correlation time denoted as T sub C instead of the birotational relaxation time. And correlation times originated in the NMR field, and it's the time for a dipole to rotate through 55 degrees, the magic angle in NMR. And the information content, though, is similar, since rho is exactly 3 times tau c. So it takes 3 times as long to go through 68.42 degrees as it does to go through 55 degrees. But it's important when you read the literature, you have to be clear if people are reporting rotational relaxation times or correlation times, because it really changes. Now, consider the case of molecules associated with proteins, for example. Like if you have non-covalently associated, like porphyrins, FAD, NADH, ANS, we'll give other examples later. If the probe is bound with any appreciable free energy of binding, it has to be held to the protein matrix by several points of attachment. So its local mobility, that is, its ability to rotate independent of the overall global motion of the protein, is very restricted. But if it's covalently attached, we'll talk about those reactions soon through an amine group or sulfhydryl group, or in the case of tryptophan or tyrosine side chains, you can have considerable local motion of the fluorophore, plus you can have domain movement. And I show this here. In this case, the protein I'd worked on, Tau7, Tau12, it's a dimer. We have overall rotation of the dimer. We can have movement of one domain, a C-terminal domain relative to other domains. And we can have movement of a dimolecule around the point of attachment. All of these will be polarized. Now, what do we use polarizations for? Well, it's very good for looking at different things. For example, aggregation state of a protein. Let's consider a protein dimer monomer equilibrium. So we have a dimer, we have a fluorophore on it, and as we dilute it, it can dissociate. Okay, so you can either follow the intrinsic protein fluorescence sometimes, or put on a suitable probe and follow this. Now, when I say suitable probe, in the case of polarization, what we really mean are probes of a suitable lifetime. That's why it's important to know the lifetime of the probe you're using for these measurements. For example, if we have a probe of a very, very short lifetime, less than a nanosecond, you're not going to get any sensible rotation of the dimer or monomer. You see very, very little change. If you happen to have a probe of a ridiculously long lifetime, hundreds of nanoseconds, which is possible, you might have complete depolarization during the excited state lifetime. So you have to choose the right lifetime. Here we see the rotation of, we show the monomer is faster than the dimer, so the polarization would be lower for the dimer to the relative to the monomer. And again, I note that the lifetime is very critical for these measurements. So you can simply make a plot of polarization or anisotropy versus concentration to look at the dissociation. Here we see fluorescein FITC labeled lysozyme, which is a monomer at all concentrations we look at. And so nothing changes. Here we see the protein L7, L12 that I sort of illustrated before. And as we dilute it from micromolar to nanomolar concentrations, we see a drop in the polarization, which is due to dissociation. Okay, now it's the change in polarization that matters to us. And for a true dimer monomer, I should go over almost 3 orders of magnitude, specifically 729-fold in concentration, to go from 10% dimer or 10% monomer to 90%. And I don't care why I get a particular polarization. Why is the polarization of the monomer about 0.15? Well, it has to do with all the local rotations involved. But that's not important, you see, when we're looking at these sorts of measurements. The same when we use it to study protein-ligand interactions. Here's a case of a fluorescent GTP, well, a non-hydrolyzable GTP gamma S binding to dynamina GTP binding protein. Before we add the protein, the anisotropy or polarization is very low because the mat probe is free in solution. When we add the protein, it goes up and up and up, and I can get a nice binding isotherm. Basically, I can get the binding constant about 8 micromolar in this case when we take into account the change in the quantum unit upon binding. So these are very easy and quick methods in cuvettes to do these binding measurements. And it's interesting to note that this approach revolutionized clinical chemistry when Abbott Laboratories introduced the TDX machine in 1981. This is fluorescence polarization immunoassays. And this was important because up until this time, immunoassays were done using radioactivities. And finally, there was recognition that this is dangerous and people wanted to get away from that. And that's where Abbott Labs came up with this machine. And the idea is to add a target, a fluorescent analog of the target, a drug, a barbiturate, a metabolite, whatever, and you have an antibody to that target, and you measure the polarization. First, you have the fluorophore bound to the target, the antibody, then you add biological fluid, blood, urine, etc., and you're looking to see if there's the target in there, and it will decrease the polarization by displacing it. This is just shown here simply. So you have an antibody to the fluorophore labeled antigen, get a high polarization, you add the unlabeled antigen, and it displaces it by mass action, and the polarization drops. So it's very easy, and you don't have to, in radioisotope work, you'd have to isolate pure, free, and bound. You don't even have to do that. This method is introduced by Danliker originally. And I want to show a diagram from the original Abbott Lab paper, first author Mike Jolie, who I knew pretty well. And we see they're showing polarization versus concentrations of gentamicin. So they used a fluorescently labeled antibiotic. What's of interest is look at the units there. And I highlight this. This was in the first paper. For convenience, they represent the units as millipolarization, where 1 MP, 1 millipolarization unit, is 0.001 P. Well, I went up there in 1981 in Chicago, where they were located, and said, why on earth do you do this? Why don't you just use the polarization? And they explained to me, well, they wanted this to be a point of care instrument, so in doctors' offices, et cetera. And they showed this to a number of doctors, and they said, well, it looks okay, but you're going from like 0.2 to 0.1. This change is so very, very small. It's not very good. So they changed it to millipolarization. Now you're going from 200 to 100. And the doctors all loved that. They said, now we have a change. So, of course, I pointed out you should have called it nanopolarization. You have a really big change. But this is why in clinical chemistry, you will almost always see these millipolarization units, and they call them milliPs. And or milli-anisotropy units, but this is very common in clinical chemistry. And you have high throughput polarization plate readers now, and even there, as you see on the screen, they're displaying millipolarization units. You can even do fluorescent polarization microscopy. I don't think we're talking in this course about that. And there are some technical issues because of numerical aperture effects. We won't go into that, but some people have done this. Here we see an anisotropy image where they're looking at different circumstances at the polarization or anisotropy of a GPI anchored proteins in cell membranes, but it's not so common. So let's move to another topic, Förster resonance energy transfer. It's named that after Theodore Förster, who made some important contributions to this. And what is FRET? Well, we have a donor molecule, we'll call it donor. There's nothing else happening. It absorbs light and emits light like any fluorophore. But if you have an acceptor molecule, which has the right characteristic nearby, we'll define what we mean by that soon, you can have transfer of excited state energy, radiationless transfer from the donor to acceptor, and the acceptor fluoresces. If the acceptor is a fluorophore, you can have a dark acceptor. You don't need to have a fluorescent acceptor. Now, I like to give this tuning fork analogy, which I learned from Anton Visser, a longtime fluorescence friend of ours. And if you have a tuning fork, we used to do this when I was in elementary school, and you hit it with a rubber mallet, it starts vibrating. Now you have another tuning fork nearby, which has the same characteristics. When that energy from that vibrating tuning fork reaches it, through the air in this case, it will start vibrating too. It has the right frequency. And this is sort of what's happening with energy transfer. You get oscillations of the electric vector in the donor, and if you have a match with the acceptor, you can get that oscillating, and then the emission can come from that. And we talk about the Förster critical transfer distance, which is the distance R0 where you get 50% energy transfer between donor and acceptor. So how far away must the acceptor be to see energy transfer? And that's going to depend on the refractive index, the quantum unit of the donor, kappa squared, the orientation factor, which is the most difficult one, which is the relative orientation of the donor of the dipoles of the donor and acceptor, and the overlap integral. You have to have some overlap between the emission spectrum of the donor and the absorption spectrum of the acceptor. And you can determine these efficiencies by looking at the decrease in donor fluorescence, for example, by intensity, or you can do it by looking at the lifetime. Then you would have the efficiency from the change in the lifetime of the donor in the presence of the acceptor. And as you learn in this course, lifetime determinations are much more robust than intensity measurements, and you're going to learn in following lectures how FRET and lifetime data are handled, largely in the microscope. What about fluorescent probes? We might ask, where do fluorophores come from? Well, some occur in nature naturally, like quinine. We're going to talk a lot about that in the history lecture. And there's many natural fluorophores, like aromatic amino acids of proteins are intrinsic fluorophores. Modern fluorescence studies would not be possible without fluorescent molecules designed and synthesized in the lab. In fact, the rapid development of biological fluorescence owes as much to synthetic organic chemistry as it does to modern molecular biology and commercialization of instrumentation. And most beginning fluorescence practitioners today don't have to synthesize their own fluorophores, since thousands are literally available now. But even as late as the 1970s, when I was a graduate student with Gregorio Weber at the University of Illinois, only a handful of fluorescent probes were commercially available. Consequently, for some of my studies, I had to synthesize the probe, build the instrument, and walk 5 miles uphill through the lab every day, through the snow every day to the lab. Okay, maybe I didn't have to walk 5 miles through the snow uphill, but the rest is true. So let's talk about probes. We talk about in vitro probes used in cuvettes or in silico plate readers, or in vivo probes, more accurately in cells. We can classify them as intrinsic fluorophores. These are fluorophores that are naturally part of the system, or extrinsic fluorophores we have to introduce into the system. Here's some pictures of intrinsically fluorescent molecules. You'll learn a lot in this course about NADH, and FAD is very common too. Porphyrins, if you take the metal out or take the iron out, you can have fluorescence from porphyrins, and there's other ones. And proteins, of course, have naturally occurring fluorophores, namely the aromatic amino acids, phenylalanine, tyrosine, tryptophan. As we see from the lower left, the absorption of tryptophan is much stronger than tyrosine and phenylalanine. Okay, and on the right, we see the emission of the tryptophan is well separated from tyrosine and phenylalanine. And that's why, and the fact that there's fewer tryptophans than tyrosines and phenylalanines usually, why people like to use tryptophan to study proteins, and especially they like to have single tryptophan protein. Now, extrinsic fluorophores, these again, as I said, are dyes you add to the specimen to introduce fluorescence. You can have non-covalent interactions that'll bind just thermodynamically bind reversibly to a specific region, like an antigen-antibody interaction, or covalent interaction, where you can react covalently with a specific functional group. So extrinsic probes, for example, some probes that bind the proteins are these famous aniline naphthalene sulfonates, ANS, we'll talk about that later, SANS, TNS. And when they bind to hydrophobic patches on proteins, the fluorescence is enhanced, as we'll see. You can have probes that bind to DNA. Probably you all know aethylene bromide and DAPI, different Hawkes dyes bind to DNA. What about covalent attachments? Well, the first covalent fluorescence probe utilized was actually Anthracene Isocyanate, made by Albert Coons in 1941 for labeling antibodies. But he was looking at serum, blood serum, for example, and interference from autofluorescence led him to prepare Fluorescein Isocyante. Now, Riggs and colleagues in 1958 described Fluorescein Isothiocyanate, you see the sulfur group replacing the oxygen, because it was easier to synthesize and it had much more stability than isocyanate. So FITC since then has become probably the most utilized probe in history. Gregorio Weber, who we'll learn more about later, in 1952 synthesized danzo chloride to attach to proteins to study protein hydrodynamics, and this initiated the field of quantitative biological fluorescence that we are in today. But it's interesting to note that one of the big increases in interest in fluorescence was due to the company started by Richard and Rosaria Haugland in their kitchen in Minnesota in 1975. Molecular probes. Many of you have heard of this. And for a brief time, they were located in Plano, Texas. And their first popular product was Texas Red, we see here, which was the sulfonyl chloride to covalently attach to proteins like antibodies. It's the big thing, immunoglobulins, to label antibodies. And that was a very popular, made them a lot of money. In the early 1980s, they moved to Oregon. And in 2003, they were bought by Invitrogen for $325 million. I understand 200 million went in their pockets. So it is true, you can become rich in fluorescence. Enrico and I haven't quite found this pathway yet, but it is possible for young people to aspire to. And an interesting series of probes they made, which has become very important, is the Alexa Fluor series. In 1999, they published this paper describing these probes, saying that there was a need for probes with high quantum yields and high photostability. That was because people were really starting to do microscopy. And fluorescein, which was a common probe, would bleach so readily. It was so non-photostable that they needed other probes. by the way, they named them Alexa after their son Alex Howland. That's why they're called the Alexa probes. So they designed a series of probes where, based on other fluorophores, coumarins, fluoresceins, rhodamines, but by making the right substituents, they introduce a lot of photostability. And here's an example they published. If you look at Alexa Fluor 488, which absorbs, emits like fluorescein, you can see it's much more stable than fluorescein is. Now, I should say there are new probes, we'll mention some later on, certainly dyes, daylight, and other probes, Janilia probes, that are even more stable than Alexa probes. So this has always been one of the goals of developing probes for microscopy, to have photostability. And here we see the Alexa Fluor dyes that go over quite a wide range of wavelengths. So you can get, this is for amine labeled things. What about labeling? Well, proteins have reactive groups like lysine residues or N-terminus NH2 amines or cysteines. And then you can put the reactive groups on your probe depending on what you want to label. And the fluorophore will depend on the light source, the spectral properties you want, maybe autofluorescence you're awaiting, etc. And of course, when you label, you want to be sure to test that it doesn't alter the biological activity. Now, Gregorio Weber and colleagues introduced polarization to study physical state of lipids and monomembranes, an idea he had in his PhD thesis. Actually, they used parylene and other nonpolar fluorescent dyes. And years later, Mayer Shininski, who was a postdoc with Weber and a co-author on that paper, with Barinholtz, introduced DPH, diphenyl-hexatriene. And for many decades, this became the most popular membrane probe. In recent years, as you'll learn a lot in this course, Laurdan has become one of the most popular fluorescent membrane probes. This was introduced by Fay Ferris and Gregorio Weber. Fay was Weber's technician, I think, in 1979. And you'll learn a lot about Laurdan in this course, so I won't go into that. But it's interesting to look at, I did a search on PubMed, and we see the DPH fluorescence papers go up and up and up from starting when it first 74 in the early 90s, and then it starts slowly going down. And it's interesting because Laurdan started going up in the 90s. It was introduced, really used by Enrico and his collaborators in the 80s. But as the Laurdan started to go up and up, the DPH starts to go down, and I think that's cause and effect. Well, there's obviously fluorophores for a wide range of ions. You can get for sodium, calcium, magnesium, anything you want, and anions. And very, very popular probes for fluorescent probes for calcium determination. I should say virtually all of them use this same theme of the 4 carboxylic acid groups, which is like an EDTA, which chelate the calcium. What about fluorescent proteins? That'll be the final part of, almost the final part of my talk. Well, as our friend Richard Day and his collaborator Davidson said in this book they edited, it's certainly been a fluorescent protein revolution. And I think you all know why, because you can take the gene for your protein of interest and attach to it, using molecular biology, the DNA of the gene for a fluorescent protein like GFP. Then when it's expressed, you have your target protein linked to a fluorescent protein. And the first one in this series was green fluorescent protein, isolated from the Aquaria Victoria jellyfish by Shimomura. And he was actually interested in the aquarin, bioluminescent compound in the jellyfish, which absorbs, which gets excited by some movement and emits blue light, which is absorbed by the GFP, which is nearby in the jellyfish. The GFP then emits the green light. Why would you want to do this? Well, because, as you know, the sky is blue, right? That's because blue is scattered much more readily than the green light. So green light will go farther. And why do they do it at all? I heard a nice explanation from a marine biologist. You don't do it to scare off the prey. The jellyfish and other things that do this do it because when they're attacked by a prey, they give off a flash of green light in the hopes of attracting a bigger prey fish to feed on the predator that's after it. So it's an alert system. Now there's been people like Roger Sen in particular and others show that you can mutate amino acids around the chromophore in the GFP and get many different colors, and there's a whole rainbow of coloring now. And I got some nice slides from Richard Day from a fluorescent course he taught. You get now, besides from the jellyfish, you get them from corals. The DS red was one of the first ones. Here we have a nice turquoise. Here's a ruby from a coral. Here's an orange, and even from fish. Here's neon green from a certain fish. And here's a nice PowerPoint from his lecture, which he credits to Adam et al., where you look at the brightness of different fluorescent proteins as a function of the emission wavelength, normalized to EGFP, the hydrogen atom of the fluorescent protein. We can see there are some much brighter and some weaker over a wide range. And you might consider this when you're choosing fluorescent proteins for your studies. How many are there? Well, hundreds. I asked Richard Day, he says, who knows, hundreds and hundreds. And he gave me this nice paper where you can look at the evolution of the green protein and how a fluorescent protein, some species lost it early on and some kept it right to the end. And humans lost it later than some others, but we did lose it. And I like to think that it's unfortunate. Think of the possibilities of fluorescent protein genes were not lost early in human evolution. But that's another story. There's other techniques like bimolecular fluorescent complementation, and Tom Kerpeler showed you can actually split the fluorescent protein into two halves and put one half on one target protein, one on the other, and if they come together, you might get fluorescence. Not always, but if you do, then you know they were together. There's other tags like halo tags, where you attach a dehalogenase to the target protein, and then you can add solution any fluorophore you want linked to the alkane that reacts, links it to the target. Similar but different chemistries are used for snap tags and clip tags. More recently, we have a series of Janelia probes made by Luke Lavis and Howard Hughes, and he developed probes that are much brighter and much more photostable, and these are becoming very popular. And I like these fluorogen-based probes by Gautier, and these papers describing these are in your PDFs, I believe. And he attached sort of a little antibody type molecule to whatever probe he wants, attach that to the protein of interest, then the fluorogen doesn't light up until it binds to that. Finally, we have quantum dots. In the 70s, Alexei Akimov first made these, and they're semiconductors. So inorganics, usually cadmium selenide or other ones. And he observed that when you get different sizes, you get different colors. So we can see on the right the different emission spectrum of different size quantum dots. And the quantum dots have been functionalized, so you can now attach them to many different molecules. And they're very, very bright, very photostable, but of course they're very large. So they're not useful for every application, but they're still becoming more and more popular in fluorescence. So at any rate, at that, I'm done, and I see I've been an hour and 1 minute, so I'm on schedule. So I'll stop.